Hello guys, this is H2O Club. In this video, we are going to talk about meiosis and mitosis. This video consists of explanation of meiosis, mitosis, the difference and similarities between the two, and an experiment related to this topic. First, we're going to talk about meiosis. Meiosis is a process that cells go through in order to produce gametes, which are sperm and egg cells. In daughter cells, we have half as many chromosomes as the starting cell. For a human, this is from 46 to 23 chromosomes. Gamete cells each contain 23 chromosomes, unlike other cells. When they come together, a newly fertilized egg containing 46 chromosomes will form. Through this process, a new genome is produced. These are the steps of meiosis. Remember, all this occurs after the interphase, where cells prepare themselves for meiosis or mitosis. Notice that the process of meiosis is divided into two big steps, meiosis 1 and meiosis 2. Both of these big steps include prophase, metaphase, anaphase, and telophase. These are the details of meiosis 1. In prophase 1, chromosomes condense, crossing over occurs, and nuclear envelope breaks down. Crossing over is the swapping over the genetic material between the chromosomes. In metaphase 1, paired up chromosomes line up in the center of the cell. In anaphase 1, homologous chromosomes are pulled to opposite poles of the cell. Homologous chromosomes are chromosome pairs that are similar in length, gene position, and centromere location. In telophase 1, the chromosomes once again gather in the center of each cell, and the cell divides. Then, meiosis 2 begins. In prophase 1, new spindle forms around the chromosomes. In metaphase 2, chromosomes line up in the middle. In anaphase 2, chromatids are pulled apart. And in telophase 2, the cytoplasm divides. Now let's talk about mitosis. Mitosis is a process that body cells replicate to grow and to repair damaged cells. Their daughter cells will be exactly the same as their parental cells, including the organelles and the number of chromosomes. Every human body cell always contains 46 chromosomes. There are steps of mitosis. Since mitosis is to produce two cells, its process is more simple than meiosis. Notice that mitosis contains only 4 stages: prophase, metaphase, anaphase, and telophase. Mitosis has no big difference from meiosis. However, each step is slightly different, so be careful not to be confused. In prophase, mitosis begins by replicating chromosomes, forming spindle fibers, and dissolving nuclear envelope. During metaphase, chromosomes line up along the center of the cell, and then spindle fibers attach to the chromosomes. In anaphase, each chromatid is pulled apart to opposite sides by spindle fibers. Then during telophase, spindle fibers disappear, nuclear envelope forms, and cells finally break apart. Here is a diagram that shows the overall process of both meiosis and mitosis. So what are the differences between meiosis and mitosis? Mitosis is the cell division of body cells, whereas meiosis is the division of sex cells. 2 daughter cells are produced in mitosis, whereas 4 daughter cells are produced in meiosis. Cell division occurs once in mitosis, whereas cell division occurs twice in meiosis. Now that we talked about the differences, what are some similarities? Both are involved in making new cells, both occur after the interphase, and both are very important for every organism. To observe phenomenon of meiosis, we conducted an experiment. Materials needed were microscope, glass slides, onion root, HCl solution, hot plate, acetylcarmine stain, razor blade, and pipette. Following were the procedures. First, we needed to grow onion roots. Then, we get 2 or 3 roots of onion and cut them into small pieces. Before the actual observation, we needed to prepare an acetyl alcohol solution of 1:3 ratio of acetic acid and ethanol. Keep the root tips in solution for 24 hours. Then place 1 drop of HCl solution on the root tip for 10 minutes. Remove it and add 2 to 3 drops of acetylcarmine stain to the slide. Warm the slide gently over the hot plate for about 8 minutes. You shouldn't allow the slide to get hot to the touch. Remove the stain with water. Then, using a razor blade, cut the more stained tip portion of the root, leave it on the slide, and discard the remaining portion. Put 1 drop of water on the root tip. Mount a cover slip on it using a needle, making sure no air bubbles exist. The root tip should spread out to a diameter of about 0.5 to 1 centimeter. Finally, observe it under a compound microscope in 10x objective. Scan and narrow down to a region containing dividing cells and switch to a 40x for a better view. Unfortunately, we couldn't observe any mitosis activity in the microscopic level from our experiment. Instead, we will show you what others got for their experiment. These are from document about cell cycle in onion root. Let's start from interface. The picture does not show the face clearly, but there are 2 nuclei in the first picture, which means the duplication occurred. Next is prophase. We can divide this phase in 3 stages: early, middle, and late. This phase shows that the chromosomes gradually condense and nuclear envelope disintegrates. In metaphase, the chromosome starts to align along the center. In anaphase, the sister chromosomes are pulled away to the opposite side. Lastly, in telophase, nuclear envelope forms again, and cell division begins. Since the mitosis happens in a plant cell, it forms cell wall and cytokinesis. This is how mitosis works in onion root and plant cells in general. Thank you for watching our video.